I hope everybody had a great weekend. No? Productive? I had a productive week. I wrote 4 new songs this weekend. Yeah. I was very happy with that. Most of them are kind of silly, though. So maybe eventually you'll hear some of them. So I forgot to post the grade distribution on the schedule page. It just slipped my mind. Somebody reminded me this morning. So I will get that done this evening. Excuse me. It's not a secret. I just forgot to post it. So I will post the distribution of grades. It'll be, like I said, it'll be on the schedule page. And you can take a look at it and see where you stand and so forth. So as always, if you have questions or anything, of course, let me know. We are slogging our way through enzyme control. And so we've seen some mechanisms so far. We saw allosteric control was a very important and powerful mechanism. You've also seen covalent modification as a mechanism that's very important. And with covalent modification, you also saw how protein kinase A was regulated in that there was basically something that blocked access to its active site. You'll see that also today in something else I'm going to show you that has some more important relevance for smokers. And I'll talk about that in just a little bit. But before I do that, I do need to finish up talking about some of the covalent modifications that occur with respect to proteins. So that's something we need to keep in mind. All right, well, I talked last time very briefly about how the zymogens are made and why zymogens were very important. I showed you this figure here, which I described as a master activation switch. I'm going to back up and show a couple previous figures in a minute that relate to this. But what this figure shows, I think, very nicely is how many enzymes, in this case, that are involved in the digestive process are synthesized in an inactive form. And as I noted last time, that was important because we don't want them attacking the tissue in which they're being made. So making them in an inactive form and then releasing them into the digestive system where they become activated by other enzymes that have already been activated turns out to be a very useful kind of a thing. One of the things that you see from this and you learn from this is the fact that the digestive system itself has cells that, not surprisingly, are existing in a pretty nasty environment. Okay? So we're protecting our pancreas by making the enzymes in an inactive form, but you're thinking about, well, what about the cells of the intestine? I mean, aren't they getting attacked by these proteases and various enzymes that are involved in digestion? And the answer is, to some extent, they are. One of the most rapidly turning over tissues in your body are your intestinal cells. So when you hear about people developing ulcers and they are taking, for example, aspirin, we'll talk about aspirin later, one of the things we've learned in recent years is that aspirin actually is blocking a group of molecules called prostaglandins, and I'm just telling you this, you're not responsible for this at the moment, that are involved in helping to synthesize intestinal cells. So aspirin actually blocks, for some people, blocks the production of those prostaglandins and stops the proliferation of those intestinal cells. So the intestinal cells have to last longer, and they don't, and people develop ulcers, and they develop stomach problems and other digestive system problems. So that's one of the examples of where knowledge of digestive enzymes actually is helpful and important. Well, you notice that there's, as you look at this, that there's a sort of a nomenclature that is given to the inactive forms of the enzymes. You see it's not consistent. All right? So typically, or at least in modern naming, the OGEN is put onto the end of an inactive form of an enzyme. So chymotrypsin is initially made in an inactive form as something called Chymotrypsinogen, and it gets activated by trypsin, and that activation by trypsin, as we will see, involves cleavage of some peptide bonds that freeze and opens up the active site so that the active site can be accessible to bind substrates and, of course, cleave proteins. We see that Trypsin itself is made in an inactive form as trypsinogen. But we also see over here, here's proelastase. What's that business? Well, in the old nomenclature, pro was the name that they put in front of the inactive form of enzymes. So you actually see both of these, the ogen on the end or the pro at the very beginning. Okay? So generally, those are the naming rules. Pro-carboxypeptidase, of course, becomes carboxypeptidase. Pro-lipase becomes lipase, et cetera, et cetera. There is a fairly elaborate scheme that's involved in activating all of these. But as I said, once it's in the digestive system, all of these are already present, and so it's very easy for any of these to become activated. Trypsin is sort of a master activator of all of the enzymes you see down on the lower part of the figure. Well, let's look a little bit more closely at chymotrypsinogen and see what happens to it. So chymotrypsinogen is initially made as a protein that has 245 amino acids in one long stretch. That's chymotrypsinogen. It is inactive, as its name suggests. And cleaving of chymotrypsinogen by trypsin creates a break in a peptide bond between residues 15 and 16. Okay? Now, this 15 and 16, this piece over here, 1 to 15, and this piece over here, 16 to 245, they don't go flying away. What would you predict would be the reason why they don't go flying away? Disulfide bonds. Very good. Okay? So disulfide bonds are strong bonds that help to hold things together. And of course, disulfide bonds aren't being broken by a protease, because proteases are breaking peptide bonds. So this guy is held into close proximity by disulfide bonds. It doesn't go flying away. But more importantly, a sort of crack in the armor of the enzyme has been created by the action of trypsin. This creates a slightly active form of chymotrypsin called pi-chymotrypsin. And pi-chymotrypsin has really one thing that it will work on, pi-chymotrypsin. So pi-chymotrypsin will cleave other pi-chymotrypsins, and when it does, you can see what it does. It puts some other breaks in here. It actually puts a break between 13 and 14, and you see that 14 and 15 don't have disulfide bonds to anything, so they go flying away. It also makes a break between 146 and 147 and between 148 and 149, and that other little piece goes flying away. And no, I don't care if you know those numbers. It doesn't really matter. Okay? But the final result of this is that the result of these cleavages is we get 3 pieces, all of which are, in fact, held together by disulfide bonds. And this last form, which is called alpha-chymotrypsin, is completely active. Now it will cut things besides other chymotrypsins. It will start cleaving any proteins that have the right binding site for it in the active site. Okay? So a several-step process that we see here that results in the activation of chymotrypsin, the net result of which is by the time this guy is out in the digestive system, it's doing its thing. Okay. This figure shows the comparison between the chymotrypsin, which is in the blue, that's the active form, excuse me, and the red, which is the chymotrypsinogen, which is shown here. So we're comparing the structures of the two, and we see that the structures, not too surprisingly, are very, very similar to each other. And what we're really interested in is, well, what's the differences in these structures? Because it explains why one is active and the other one is not. Well, one of the critical cleavages that occurs is right here, isoleucine number 16. Okay? And when that gets cleaved, we see that this big red piece that's sticking up here isn't present in the final chymotrypsin because we don't see a blue piece up there corresponding to it. This red piece up here serves to help block access of the substrate to the active site. So, by opening this up, we start to allow access. Other places where we get cleavages are shown in here. And the consequence of which is ultimately the enzyme becomes fully opened up. But the very first thing that has to happen is we have to release this little piece up here so that the substrate can get to the active site. And that happens by cleaving isoleucine number 16. And again, I don't care that you know the numbers. I'm just illustrating the principle and showing to you what the difference is between the enzyme in the one form and the enzyme in the other form. Questions about that? Monday. Nobody wants any questions. Okay. All right. Let's see. So that talks then about the activation of the proteases, which are synthesized as zymogens. I want to say one other thing about proteases that are relevant to the topic of proteolytic enzymes. And this actually relates to your lungs. Okay? So your lungs have an enzyme in them called elastase. Okay? And elastase is a protease. And elastase is very good at breaking down proteins. And that turns out to be useful in your lungs. You might wonder why that's useful in your lungs, but part of the reason it's useful in your lungs is because you breathe, which is what your lungs allow you to do. And when you breathe, what do you get in from the environment? Well, you get bacteria, you get viruses, you get all kinds of junk that's coming in there, and you'd kind of like to get rid of it, right? Well, elastase is one of those enzymes that is pretty good at at breaking proteins down in these various things that we breathe, and it helps to reduce our likelihood of getting infections in our lungs and so forth. Why do I tell you that? Well, I tell you that because there's an inhibitor of elastase that's known as alpha-1 antitrypsin. That's kind of an odd name for something that inhibits elastase, but I'll tell you the story of it. So alpha-1 antitrypsin is a protein. It's a protein that is very good at plugging up the active site of trypsin. So when they discovered it, they called it alpha-1 antitrypsin because it would bind to the active site of trypsin, and trypsin would consequently not be able to do any more proteolytic degradation. It stopped it. Okay? The similar thing that we saw that the regulatory sites, the regulatory peptides of the protein kinase A blocked the active site of protein kinase A, the catalytic subunits of protein kinase A, so that it wouldn't work either. So physically blocking that active site was a very useful approach to controlling the enzyme. How does that relate to elastase? Well, when they studied alpha-1 antitrypsin more carefully, what they discovered was it actually works really well on elastase, and it might even work better on elastase than it works on trypsin. But the name was already there, so they call it alpha-1 antitrypsin. All right? Well, we have alpha-1 antitrypsin in our lungs along with elastase. That's kind of odd. Why do we have that? Okay? Don't we want to have elastase going along and breaking down these various proteins and so forth that we're getting in our breathing? The answer is yes, we do. But just like our intestinal tissue, or just like our pancreas, we also don't want elastase attacking our lung tissue. So alpha-1 antitrypsin turns out to be a sort of a buffer, okay, for elastase, meaning that you make it in the right quantities, you have about the right amount of elastase that will be active. You make too much, you won't have enough elastase active. You make too little, you'll have too much elastase active. Make sense? Okay? So, the critical thing then is how much alpha-1 antitrypsin I have in my lungs that can block elastase. All right? Well, if I have too little alpha-1 antitrypsin, it means I have too much elastase. And what happens when I have too much elastase is elastase attacks my lung tissue, and it causes a disease we know as emphysema. Okay? So alpha-1 antitrypsin, if we have too little of it in our lungs, we will have elastase attack our lung tissue. We will develop emphysema. Okay? Now, guess what? Smokers, when they smoke, the reactive oxygen species in their smoke will oxidize a methionine in alpha-1 antitrypsin. It will oxidize a methionine in alpha-1 antitrypsin. What does that do? Well, the methionine is actually involved in binding to elastase. So the more you smoke, the more elastase you will have active, because the less alpha-1 antitrypsin that you have that functions you will have in your lungs. Now you know why smokers get emphysema. Okay? So I'll repeat that. That's kind of a convoluted sort of thing. All right? So people who smoke have what are called reactive oxygen species produced. Those reactive oxygen species will oxidize things. One of the things that they will oxidize is a specific methionine in alpha-1 antitrypsin. Okay? As a consequence, the amount of alpha-1 antitrypsin in the lungs is reduced, at least the functional amount of that is reduced, which means that the amount of elastase that's active will go up. The elastase attacks the lungs, and consequently, the lungs develop, or the person with this develops emphysema. Yes, sir? How does the body regulate alpha-1 antitrypsin? It's a very good question. It's complicated, and I don't have a simple answer for that. But needless to say, this is interfering with the body's normal regulation of alpha-1 antitrypsin. And yes, are there people who have genetic diseases who are more prone to having emphysema by having a problem with alpha-1 antitrypsin? The answer is yes, there are. Okay, questions about that? Other questions about that? Monday, you get few questions on Monday, I've noticed. Everybody's just still kind of hanging out there from the weekend, I think. Okay. Let's see. So that's that. Here's the methionine oxidation that actually occurs. There's the methionine residue. And we can see that this sulfur has just a simple oxygen that's added to it, but that's sufficient to stop or significantly reduce the binding of alpha-1 antitrypsin to the elastase, and consequently the elastase is left in a more active state. Okay. Well, so we've turned. Now I want to turn our attention away from protease, or at least the digestive proteases, and talk about a different group of proteases that are probably equally fascinating. And these are the proteases that are involved in blood clotting. Okay? Blood clotting is a pretty essential thing for a multicellular organism. All right? If you think about it, and we sort of take it for granted, but if you think about what has to happen, all right? If I go out and I cut my finger, okay? If I don't have something to stop the blood flow, I will bleed to death. Now, we take it for granted that we don't have to think about that because we have a clotting process that takes care of that. But it's important to remember that. We've got a clotting process that takes care of that, okay? We can even, repair and stop the blood flow for fairly serious types of cuts that we have. Now, if we think about this for a more serious type cut than, say, cutting my finger, we realize that the challenges that the body has in trying to manage blood flow. All right? So, for example, when I cut, let's say, a major artery, all right, then my body has got to very quickly put something in place to stop that blood from exiting the body and keep it in the system that's there. Okay? It has to have a system that is ready to start literally at the drop of a hat, and it needs to assemble a repair, or at least a temporary repair, within minutes, because if not, you're going to bleed to death. And, more importantly, that repair system should only be activated by an injury. Because we don't want it forming clots when there's not an injury, because clots traveling through our bloodstream hit our brain, hit our heart, cause heart attack, cause stroke. Okay? So this is pretty remarkable. Excuse me. This is a very big demand that we've placed on our body. It means that we need to have floating in our bloodstream the ability to form a clot very quickly, a hair trigger, but that hair trigger only fires when we want it to fire. Okay? Now, to do this, the cell accomplishes this by using a cascade. You saw the cascade that I showed you for the proteolytic enzymes in the digestive system. The enteropeptidase activated the trypsin, and the trypsin activated the chymotrypsin. Okay? If we think about that enteropeptidase that converts trypsinogen into trypsin, it might be acting, enzymes work on a few substrates, right? So it might activate, let's say, 1,000 trypsinogens to become trypsin. But now those 1,000 trypsins can activate 1,000,000 chymotrypsinogens to become chymotrypsin. So at each step in that cascade, what we see is an amplification of the signal. And that amplification buys us time because it means this mobilization, this change that happens, can happen very, very rapidly. Okay? Well, it turns out that works out very well for our bloodstream because we need to have that bloodstream working for us very, very rapidly. Okay? Here's what the bloodstream cascade looks like. Now, no, we're not going to memorize this. All right? But I show it to you to show the complexity of what's involved in making a blood clot. The blood clot is formed down here at the bottom. Okay? Here's where the blood clot forms. All right? It forms as a result of the self-assembly of protein subunits called fibrin. Okay? When you have fibrin present, they will self-assemble and they will form a structure that ultimately will hold water. It will be water resistant. Okay? This is nothing short of remarkable. And it does it in seconds or minutes. All right? All right. So here is our goal. We want to get to here. All right? Now, there's a lot of complexity here, and as I said, I'm not going to hold you responsible for the complexity. I will talk about a few components of it because I think they're important for you to understand. Okay? Fibrin's going down to make this clot down here. It's also, you can see these various factors that are involved in helping this process to occur, and you can see various proteases that are involved in converting an inactive form, which is in red, to an active form, which is in yellow. Okay? So the active forms are in yellow, the inactive forms are in red. Well, if we start at the very top up here, we see that there's a cascade that happens. We can actually activate the clotting process by two different pathways, and I'm not going to talk about what they are, but two different pathways can lead ultimately to a blood clot. And interestingly, the two pathways converge at a point, meaning I could start over here and make a clot, I could start over here and make a clot, or I could start at both and make a clot. Okay. So who are the players here? Who are the players? Well, let's imagine starting up here, all right, at the very top. At the very top, we have one enzyme that is activated. It's activated by damage, all right? That one enzyme makes 1,000, which makes 1,000,000, which makes 1,000,000,000, which makes, okay, you start seeing again a very large amplification of that signal, and that amplification is happening, as I said, very, very rapidly. If we look what it is that converts, well, that makes fibrin, all right? Fibrin starts out as an inactive form. It's called fibrinogen, and our bloodstream is loaded with fibrinogen because we like to be able to make clots as quickly as we can when we need to. Fibrinogen is the inactive form. It's converted into fibrin by action of a protease known as thrombin. Okay. Well, thrombin, not surprisingly, is also made in an inactive form called prothrombin. Okay? Prothrombin is converted into thrombin by this converging protease up here, XA, all right? 10A actually is what it is, 10A. And that causes this bottom part of the pathway to occur. Okay? We're going to focus essentially all of our attention down here on this lower part. Because for our purposes, this is where the real action is going to be. Okay? So, we're interested then in the conversion of prothrombin to thrombin, fibrinogen to fibrin, and the formation of a blood clot. The blood clot is made initially as a very loose structure, and then we'll see how it gets hardened with action of an enzyme. And you probably have seen this with clotting on your own skin. the initial thing that will form will be a very loose kind of clot that you can bump and cause it to bleed again very readily, but you give it a couple of hours or you give it a few hours and you get something that's dry and that is much more stable and solid, and that happens as a result of hardening of the clot that we'll talk about. Okay, so let's look and see what's happening here. All right, so we're going to start down talking about fibrinogen. All right, and fibrinogen looks like this. Okay, fibrinogen has 3 chains associated with it. All right, they have an alpha, a beta, and a gamma. 3 chains. We see that it's a dimer. Here's another alpha, beta, and gamma. And we see that the alpha, which is this red guy, has a structure on the end of it called capital A. It's a good name for it, capital A. All right? And we see that the beta, which is shown here in blue, has a structure on it called capital B. Simple to remember. Alpha is A, beta is B. All right? What thrombin is doing is it's clipping off so that the capital A and the capital B disappear. We're left with the little skinny part, but the fat part is disappearing. Okay? Everybody with me? Now, that removal of the capital A and the capital B is sufficient to cause the polymerization to start to occur. Because the A, what's left over of the alpha, where the A was, this little skinny part right here, the skinny part of the alpha, can fit into this part of the gamma. Okay? So we see this little red guy can actually link to one of these, and this can link to another one of these. And we can imagine now that we can make an entire sheet of these by these guys linking inside of that little unit right there. Does that make sense? So the alphas are going into the gammas and forming a loose structure. All right? Well, what about the betas? Well, it turns out that the betas can fit into the whole of beta structures. There should be one over here as well, but they can fit into the holes of these beta structures. And now we get a 3-dimensionality of it because it's coming back out at us. So the polymerization that's happening is linking this dimer to another dimer, to another dimer, to another dimer, and it's occurring not only in the plane of the board, but it's also occurring this direction as well. All right? That's happening solely because the A's and the B's got cleaved off. Yes? They're interacting right here. Yep. Yep, you bet. Okay? So this structure gets bigger and bigger and bigger. Well, if we think about it, that's what really is happening. If we were to look at any individual protein, that would be any individual dimer that's there, it would be way smaller than we can see even with a microscope, even the most powerful microscope. We wouldn't see an individual one. But this structure can polymerize and grow thousands and thousands and millions of units to the point where we can actually see a scab. We see that clot that forms. That's really remarkable. A really remarkable thing. Okay? Now, how does the clot harden? Did you have a question?
Yeah, I was wondering if the alpha and capital A go to the betas and gammas of the same fibrin or that they originated?
It doesn't matter. All they have is, so his question was, do the A's, the alphas go to the gammas and betas of the same one? And the answer is ideally not, because what happens is the more they stretch to other ones, the longer it's going to grow. So if this guy only went to here, then we would sort of form a block within itself, but that wouldn't make a polymer. Make sense? Okay. All right. Now, what causes the clot to harden? Okay. Well, actually, let's see. Before I get to that, let's look at a polymer. So here's a polymer. You don't have to envision it. You can see now how we have these guys that are sticking together. Okay. There's the A's with the gammas. The ones coming out at you, you don't see because they can't really draw it in 3 dimensions. But we see all these various holes for the individual betas. The beta blues are going to stick in those and now come out in this direction. Okay? Okay. Hardening of the clot occurs through covalent modification. Here comes covalent modification again, guys. You know, what does covalent modification do? It gives you a strong bond. That theme has come through in disulfide bonds. It's come through in several things we've talked about. But that covalent modification gives strength to the clot. Okay? How does it work? Well, there's an enzyme called transglutaminase. And transglutaminase will catalyze a reaction between glutamines and lysines that happen to be close to each other. Okay? Is there a specific one? No. All right? Are there many glutamines and lysines that in this mess of protein that's in this polymer, are there many of them that are close together? The answer is yes, there are. So now we're forming covalent bonds between the individual dimers that are there, and those covalent bonds give this unit strength. So the hardening of the clot is made possible by the action of the enzyme glutaminase. I'm sorry, transglutaminase. And it's making this reaction right here. Okay, so that's cool. All right, questions about that? Yes? Okay, good question. How do we keep transglutaminase from acting where we don't want it to act? Well, as we will see, many of these enzymes are localized pretty close to where the clot itself is forming. I'm going to give you a mechanism by which one of those gets localized in just a second. Okay, but good question. We don't want these things going anywhere besides the clot. This question is very good because we don't want all this process happening elsewhere because now we're going to start taking all of our proteins and putting those together. We don't want that, right? Okay. All right. Actually, since I'm on the topic, let's jump to that and I'll come back to the proclotter and anticlotters here. All right? So, prothrombin turns out to be a really interesting protein. Prothrombin. Now, we're talking about the inactive form of thrombin. Prothrombin. All right? The inactive form of thrombin gets chemically modified by an enzyme that uses vitamin K. Vitamin K is an enzyme that's associated with blood clotting. It's very important for blood clotting, as we will see in a minute. This enzyme that uses vitamin K converts the glutamic acid side chains of prothrombin into something that has 2 carboxyl groups instead of 1. Normally, the side chains of glutamic acid have a carboxyl group. The R groups have a carboxyl group. But this modification that uses vitamin K adds a second carboxyl group to the side chains of glutamic acid of prothrombin. It works on other enzymes as well, but prothrombin is the one I'm going to talk about here. Everybody with me? So now we've got prothrombin, and this is activated in the area of a blood clot. This enzyme that does this is activated in the area of a blood clot. It causes the prothrombin near that clot to get an extra carboxyl group, and that chemical modification causes prothrombin to bind to calcium. It causes prothrombin to bind to calcium. Well, guess what's abundant at the site of a wound? Calcium. How is it abundant? Well, normally our cells take calcium and they sequester it inside of our cells. They sequester it because calcium is pretty nasty for our DNA. When you cut or you slice something, you're ripping open a lot of those sequestered calciums, and here's a target. All right? So now you've got calcium at the site of the wound, you've got prothrombin at the site of the wound, and now prothrombin is binding to calcium that's right there, And guess where it's going to stay? It's going to stay right there at the site of the wound. And it's going to get converted to thrombin. And thrombin is going to convert fibrinogen to fibrin. Really cool stuff. Okay? And it happens because of vitamin K. Without vitamin K, that process doesn't happen. Prothrombin doesn't get held at the site of the wound. And prothrombin doesn't get activated to become thrombin. If I can block the action of vitamin K, I can create an anti-clotter. Right? If I block the action of vitamin K is necessary for that modification. That modification holds it at the site of the wound. It means prothrombin is not going to be held at the site of the wound. And without prothrombin at the site of the wound, I don't have thrombin. I don't have fibrinogen being converted to fibrin. I've just created an anti-clotter. Okay? I'm sure all of you have heard of blood thinners. Blood thinners work on that principle. Here's vitamin K. Here's some modified forms of vitamin K. One's called dicumarol. You also hear it called dicoumarin. And warfarin. They resemble vitamin K. The enzyme that does this modification grabs one of them, except for it can't carboxylate glutamic acid residues in prothrombin. These guys are anti-clotters. They keep the enzyme that makes that modification from binding to vitamin K. Would you call them competitive inhibitors? The answer is yes, you would. Okay? Because they resemble the normal substrate. Maybe they're not perfect, but they do resemble the normal substrate. And they compete with the normal substrate, vitamin K, for binding by the enzyme. Okay? Warfarin has a more important name. It's known as rat poison. Okay? And the next time you use rat poison, this is what always happens. I tell people, imagine what you're doing to those little rats. And then everybody thinks, oh my God, they're going to bleed all over my house. Right? Well, it turns out that they don't. You get too much of this, what happens is most likely they hemorrhage internally, brain-wise, which is why you have to be very careful giving people blood thinners. And yes, they do give warfarin to people. Okay? If you give too much, then you will hemorrhage, meaning that you will have uncontrolled bleeding in your brain, and you will suffer a stroke and die from that. Okay? So people who are on blood thinners have to go in periodically to check their level of anti-clotters to make sure it's in the right acceptable range, because if not, they're at risk of hemorrhaging and having problems. Everybody's got sort of ghastly looks on their faces right now. Okay? Yes? If you were on warfarin, would you take a vitamin K supplement? No, because what you're trying to do is you're trying to block vitamin K in the first place. So adding a vitamin K back would be like adding two drugs where you didn't. Yeah, it would. So more importantly, you're trying to add the right amount of warfarin to control the amount of vitamin K. Yes? Did I say that vitamin K goes in the active site of the enzyme? I did, yes. Yes? Is any of this similar to snake venom? No, snake venoms tend to be neurotoxins. And so they nail you by nailing your nerves. They're not very, very fun. Yes? Shoot us.
Question about taking vitamin K, taking warfarin. I've actually heard in a physiology class that some people that do, get into rat poisoning, or their children get into rat poisoning when they take them to the emergency room. And
Yes.
The massive amount
Yep.
of vitamin K.
Yep.
And,
Yes. So what Shanice just said is true. People who, for example, ate rat poison without realizing it, or children or something like that, and they had a very big dose, you don't want them to hemorrhage. So in that case, giving them vitamin K might make some sense. One has to be careful with this because, again, we have to control these levels very carefully. If we add too much vitamin K, now we've got the opposite problem. We may be forming clots where we don't want clots happening. So that would be given very, very careful and controlled doses, but I have heard of that being done, yes. Yes? What triggers the vitamin K to bind to the enzyme? It's the substrate. It's a substrate. So just like any other substrate will bind to an enzyme, it's a substrate. Okay, so he's asking, how do we control, how do we get, so I can't go through the whole clotting process with you, but suffice it to say that these are controlled within the region of the clot fairly well. Okay? When you cut yourself, you're not more at risk, for example, to forming a brain blood clot. Okay? So there are controls that are there that help protect you, but it's more than I can go into here. Yes? How does hemophilia relate into all this? Yeah, hemophilia is a genetic disease where people are missing some of the factors that are necessary in that cascade. So if you can't, let's say you have a cascade that starts, but it's missing a factor, it can't jump to the next one, then that person will never be able to activate prothrombin to become thrombin, activate fibrinogen to become fibrin, and therefore will bleed to death. So people who have hemophilia, that's in fact what will happen. Yeah, nasty. Yes?
Does that mean that up until the point where wherever they're missing something in cascade, where there's a hole, they get a buildup of the immediately preceding product?
So our question is, does that mean if you were missing one of the things in that cascade that you would have a buildup to that point and then nothing beyond that? And the answer is yes, you would. Yeah. Okay, good questions. Okay, blood clotting always stimulates a fair amount of discussion like that. Actually, I didn't show you the modification. There's the modification. There's the second carboxyl group that gets put onto there. And the beauty of the second carboxyl group, I forgot to mention this, is that it's right next to the other carboxyl group, and that dimension right there is exactly between those two minus signs right there, is exactly the dimension of a calcium ion, which is charged +2. Very, very cool modification. If you don't have this carboxylation, the calcium binding is very, very weak by the prothrombin. Very, very weak. Okay. Well, we talked about how we can make clots. How about breaking them down? All right? We do have to break down clots because our body puts a clot in place for the healing process to go on, but the body also wants to break clots down as the healing process continues. All right? Well, there's an enzyme that is involved in that. Okay? And the enzyme that's involved in, actually, the enzyme that's involved in that is called plasmin, Plasmin. Plasmin has a very useful property. It's a protease that breaks down blood clots. It's a protease that breaks down blood clots. Plasmin, as you might expect, is also made in an inactive form, because we don't want it floating around all the time breaking down clots when we need them. Plasmin is made as an inactive form called plasminogen. And plasminogen is activated by an enzyme called TPA. TPA is known as tissue plasminogen activator. TPA, you can call it TPA. Right? This is schematically what it looks like. You can see it's a serine protease. So the presence of TPA will cause plasminogen to become plasmin, and plasmin will then attack blood clots. This turns out to be very useful in certain medical circumstances. Here is a remarkable photograph of a blocked artery in a heart that was treated with TPA, and you can see the blood flow after. There's before and there is after. Okay? It must be used under very controlled circumstances because, again, we have to think about we don't want every single clot to be dissolved. We have things that are protecting us in our brain and so forth, for example. But under very controlled conditions, it can be very much of a lifesaver. In some cases, it's used to treat people who have a stroke, and if they're treated very quickly with TPA after a stroke, they can sometimes regain function. So it's a very important enzyme. Okay. Let's see. Questions about that? Would you like a song to summarize it? Of course. Okay. I have a song called Thank Goodness My Blood is Clotting. And I can't sing it very well, but I have someone who will. lace on. You can sing along. I can't sing. I can't sing this worth a crap. There's some closure I need. Can't hear you. So the body kicks in, gets defenses. Many times before. Untangling vibrant chains. This calls for some problem solving. There's a way to activate of some TPA, the plasmin activating thing. And thanks to the descent clotting, as part of repairing things, it's part of my body's clotting. The wound is gone. I'm back where I started and nothing's wrong. My blood flows normal again. All right, guys.